I'm very excited. I'm very excited to introduce our third speaker of this session, Dr. Lucina Uddin. Dr. Uddin is a professor in psychiatry and behavioral sciences at UCLA, her alma mater. She's the director of the Brain Connectivity and Cognition Lab, and her research examines the organization of large-scale brain networks and their associations with social cognition and executive function. So I'm excited to turn it over to you, Lucina, and I'm looking forward to your talk.
Great. Can everyone hear me all right? Sound is okay? Okay, great. Then we'll go ahead and get started. I guess it's a 15-minute talk or so I've been told. So I just wanted to share some of our most recent work that just came out this summer that I thought might be of interest for this group. And basically it came out as a preprint a few months ago under the title, A Parsimonious Description of Global Functional Brain Organization in 3 Spatiotemporal Patterns, and just came out over the summer, and the news and all that articles are out. So if you want the abridged version, that's the best place to find it. But basically, we were starting with the observation that many of us have had over the years that, of course, the brain shows a high degree of spontaneous activity that you can see with EEG or fMRI or many other neuroimaging modalities. And that spontaneous activity isn't just random or noise, but it seems to be quite structured in large-scale brain networks. Even here at the very low frequencies, you can see coherent activity across systems that we've identified in cognitive neuroscience that are related to language and memory and attention and various other cognitive processes. So we know from the first paper published using MRI to look at this phenomenon by Bharat Biswal in 1995, that we can see these coherent patterns of activity in the brain. And since then, we've been really struggling to find ways to characterize this spontaneous activity and make sense of it. And so I wanted to introduce some terminology that many of you might be already familiar with, but we talk about different methods and approaches for studying spontaneous activity in the brain as measured with magnetic resonance imaging. And some of the approaches that are commonly used are referred to as zero lag synchrony approaches. So these are approaches that compute instantaneous statistical dependence between any 2 time courses. And things like seed-based correlation, functional connectivity is one example of a zero lag synchrony approach that looks at instantaneous correlations between time courses of 2 regions. There's also been recent work looking at gradients of functional connectivity in the brain by Margulies and colleagues. It's also an example of a zero lag or instantaneous connectivity approach. And others are going under the umbrella of time lag synchrony, which look at statistical dependencies between 2 time courses, where one is delayed in time. So some examples of these types of approaches are lag projections from Mitra and colleagues, and quasi-periodic patterns that were introduced a few years ago by Shella Kilholz and colleagues. So we know that when we're analyzing spontaneous activity in the brain, it's not as straightforward as when you're analyzing responses to task stimuli or evoked responses, because there's nothing to model from the external world. So what we have to do is come up with some statistics or come up with some assumptions about how to best characterize the activity in the brain without these a priori models. So that's why there's been sort of a proliferation of approaches in the last 20 years, and sometimes making sense of them can be challenging. So this is work by Taylor Bolt in the lab, who was a graduate student who completed his PhD in 2018, went on to do a postdoctoral fellowship at with Shella Kilholz's lab, and shortly after that joined industry and started working as a data scientist, where he's currently still employed. And actually on the side, he works as a statistics consultant for our lab. So to answer the age-old question of academia or industry, Taylor is a good example of why not both. So what he's been doing in his spare time is thinking about how to unify some of these approaches for studying spontaneous activity. And the idea was that a lot of these widely used approaches that comprise both zero lag and time lag representations of functional brain organization, captured different aspects of a small number of patterns that exhibit both zero lag and time lag structure. So the sort of specific hypotheses here are that global patterns of zero lag and time lag synchrony may be describing the same underlying spatiotemporal patterns, and that a small set of these patterns might be able to explain a large number of findings in the literature when you look more broadly at all of the work that tries to describe spontaneous blood oxygen level dependent signal fluctuations. And this is the kind of analogy we like to use when talking about this, is that the field so far has been trying to explain this phenomenon of what's the organization of the brain and how do these spontaneous signals kind of emerge. And we've all been looking at it from the viewpoint of whatever the approach is or whatever the method is that's our favorite approach. So we might get a good picture of that part of the signal, or here in this case, that part of the elephant, but still sort of lack a bigger picture understanding of the whole in this case. So this is my elephant analogy. When this paper came out, we got a really nice summary of it from Gonzalez Castillo, who inserted this really nice diagram, which we should have included in our original paper. This is a really nice demonstration of what we're trying to do here. So the idea is what Taylor did was borrow a concept from geophysics and other sciences. The idea that you can look at zero lag and time lag approaches to connectivity. You can characterize them using traveling waves and standing waves are these two concepts. So traveling wave is like what you see here, sound, kind of travels along. And a standing wave is the example here is a jump rope that kind of is a stationary wave. And there's ways of sort of formalizing these kinds of approaches that have been used in other sciences. And we basically adapted them here for looking at a neuroimaging time series. And so one approach that Taylor decided to adopt here is called complex PCA, or complex principal component analysis. And this will take a whole of 15 minutes to describe. So instead, I'm just going to give a summary that to capture both standing and traveling wave behavior of the signal in a single latent representation, what PCA allows you to do, it's a extension of PCA essentially, where you basically apply PCA to the complex bold signals. They're obtained by a Hilbert transform of the original bold signal. And it's been referred to by other names in different fields. So that's why it's actually been out there for a long time. But someone on Twitter asked about, doing a Twitter thread on complex PCA. So we tried to put this thread together, and you can refer to it later on if you want a little bit of the history of where this came from and how it's applied in the current study. But it's a way to sort of pull together both in that one latent structure, sort of the traveling and the standing wave phenomenon in the brain. So the first thing that Taylor did was create some simulations. And the idea was to sort of test the idea that a relative mixture of standing and traveling wave behavior and cortical bold signals can help us explain why we see such similarity between the outputs of zero lag types of analyses and time lag analyses. So in this kind of fake data or simulated data, he was able to show that zero lag methods mostly capture most of the variance of spatiotemporal patterns with moderate traveling wave behavior. And for systems that consist predominantly of standing waves or just a moderate degree of traveling waves, we would expect solutions of time lag and zero lag functional connectivity methods to converge. So this was a way of starting with where we knew the ground truth and seeing how much basically the different, how much CPCA could explain these phenomena that we already understand because we created these time series. And so then what he went on to do was look at empirical bold data. So this was using a subset of the Human Connectome Project data. I think it was 50 subjects, adult subjects, and then a whole replication of, in 50 other subjects. And this is the CPCA applied to the HCP resting state fMRI data. And what that showed was that there's basically three principal components that explain the majority or explain much of the variance in the signal. And after that, the sort of, if you use some metric to look at which principal components explain the most, it was the first three. And so the first component here, which we creatively call pattern one, explains about 20% of the variance in the signal. Oops, is this not going anymore? There we go. Whereas the second component, which we call pattern two, explains about 6% of variance in the overall signal, and component three explains about a little bit less, like about 5% of the variance. And so this is sort of, and the first component, of course, if you look at it in terms of its spatial layout and its sort of emergence over time, it resembles a lot of what people have been calling the global signal in the brain. And I'll get back to that a little bit more at the end of the talk. The second pattern looks a lot like this task positive, task negative network, sort of the difference between the default mode, for example, and frontal parietal networks, and that sort of representation. And so think, oh, I'm already running out of time. So let's get on to this is a little bit in the paper about the form and the properties of these three spatiotemporal patterns. So there's phase delay maps here that tell you a little bit about temporal ordering of the patterns. And so I think I just won't get into details about this, just to give us time to look at some of these other findings. And what's interesting is, if you compare these three spatiotemporal patterns that you get out of the CPCA method, they, first of all, the patterns are predominantly composed of standing waves. And this suggests that the zero lag functional connectivity methods should capture most of the variance in these patterns. So what Taylor then went on to do was use a lot of the methods that we see in the literature. So kind of the regular PCA, PCA with Baermax rotation, eigenmap, spatial ICA, temporal ICA, hidden Markov model, seed-based correlation, co-activation pattern analysis, and applied these approaches to the original time courses from the same resting state fMRI data, and then tried to match some of the patterns that come out of those approaches with the approaches he got from this new CPCA, and compare sort of the spatial topography of each of the outputs of those commonly used methods with the first three PC maps. And this is just to show a strong degree of spatial similarity between one or more of the first three PCs that came out of the newer approach, and shows basically how much resemblance there is between what you find from all of these mixed bag of approaches that people have been using. So they can all sort of be closely related to one of these three principal components. And in particular, this pattern two, which I referred to as that task positive, task negative pattern, which also looks a lot like the primary functional connectivity gradient that's been published before. So basically what we see in pattern two, that pattern that explains about 6% of the variance in the signal, is what others have described basically as the task positive, task negative, or principal FC gradient. It's just it's basically pattern two is those same phenomenon by another name. And so this is just kind of showing some of the spatial overlaps between the different methods, just to drive that point home a little bit. And this is sort of the tricky part, is what does it all mean? So the first pattern, the first principal component that explained more than 20% of the variance, which we called pattern one in this paper, is basically the same. It's like 99% overlap with the global signal, or what's been called the global signal in resting state fMRI. And it's very similar to what Shella Kilholtz and colleagues have called the quasi-periodic pattern that her group has characterized very well. So we basically are showing this kind of three canonical spatiotemporal patterns that recapitulate major findings from a wide range of other analytic techniques that have been put forth. And these patterns account for a lot of the large-scale structure that underlies functional connectivity analyses, affecting everything from functional networks to how graphs are interpreted, for example. And so, we just think that it's kind of nice to see all this convergence, but also to see like one way of unifying all the different approaches that have been put forth in the literature. And so the question of, what does it all mean? Like, what are these signals that we're seeing in the brain? It's hard to tell from fMRI data, which is noisy and has a lot of issues that we all are well aware of. So what Taylor is currently working on in an ongoing collaboration with a bunch of people is looking at multimodal, single and multi-echo fMRI data sets, EEG fMRI data sets, simultaneously recorded EEG and physiological signal data sets to look at co-fluctuations between these bold signals and a lot of physiological things like, in particular, spontaneous ventilation, so respiratory rhythms. And the dominant spatiotemporal pattern, that pattern 1 of the global signal we just saw, really does tend to correlate with changes in ventilation, and basically there's a lot of other electrophysiological signals that he's finding that are interestingly related to this overall pattern. So this is the sort of the future and current directions of this work. And I think I might be pretty close to on time. So this is just a really collaborative project between Taylor and my lab, and Shella Kilholtz at Emory, and Daniela Stock in McGill, Katie Chang over at Vanderbilt, Thomas Yeo in Singapore. And it's really been a struggle to try to figure out, what are all these signals mean now that we have them. So I'm very curious to hear anyone's thoughts and have a good discussion. Thanks.
Thank you, Lucina. That was a great talk. I'm going to open up the floor for questions, but I want to encourage more folks on Zoom. If you'd like to ask a question, feel free to raise your hand or type in the chat and I'll read it out. So does anybody have any questions for Lucina?
Hi, Lucina. It's Todd Woodward.
Hi, Todd.
How are you doing?
Good.
Good. I just wanted to tell a little anecdote and then I ask the question. The anecdote is that in my other life, not brain imaging, I'm involved in delusions and schizophrenia. And a colleague of mine and I developed a treatment for delusions of schizophrenia called metacognitive training. And that's been translated into 33 languages and it's used around the world. And halfway through this, we found out that someone else had developed a different treatment that was also called MCT, but it was called metacognitive therapy. And this led to kind of a very big controversy, a lot of letters back and forth, and actually was very uncomfortable. So you have now presented a way to investigate brain networks that's called CPCA. I have been doing investigation of brain networks with CPCA for 15 years. So we're kind of in a situation now where we can avoid the confusion potentially for people starting now, because this is the first I've heard of this and we've had contact over the past year, fair amount. I mean, I present in your lab and things like that. So what do we do?
Sorry, I'm trying to overcome the noise here. Are you saying your lab has already been using this and we're not acknowledging that? Is that what you're saying?
The thing is, it's confusing for people because we're doing a different thing. It's not complex PCA, it's constrained. But it's CPCA.
I see what you're saying.
The networks and bold signals. So today we have a chance to figure out what we can do. Confuse the heck out of people. Because when people say CPCA for brain network,
yeah,
I have no idea whether it's your CPCA or my CPCA.
No, this is a wonderful point. Thanks, Todd, because that's exactly the problem. I think we've had a little bit of proliferation, not just of methods, but of acronyms and of it's just hard to keep everything straight. So, I'm happy to use one of the other names to talk about the method. I think it goes by many, many, many names in other literatures. I think Taylor talked about it in the methods section, but I think it's called in the climate sciences, I think it's called complex orthogonal decomposition or something. I think it just goes by different names. So I'm happy to change it.
Well, that solves the problem.
Makes it any clearer.
You could make the C lowercase.
Yeah, no, it does become an issue when, these things proliferate and then people don't know what exactly it is we're talking about. So thanks for pointing that out.
Okay, we have a question on Zoom that I'm going to take. Norman, you want to go ahead?
Hi, this is Norman from Michigan State. It's so awesome that you're doing this free that everyone can join. I just have a short question. This was very interesting that you found this correlation with breathing in the main first component, basically, that you find. Did you also look at like cardiac components and of like fluctuation of heart rate and these things?
Yeah, this is a great question. This is exactly what Taylor's working on right now. He's got these all these signals that he's correlating that are from these multiple data sets. And I think the short answer is yes, I think he's also finding that. I just gave one quick slide at the end that shows one of the hundreds of analyses that he's currently working on. But yeah, the idea that we're actually finding is that people are quick to, dismiss respiratory and cardiac and all these other signals and call them noise. But the truth is they co-vary with a lot of the brain function, right? I mean, they have to. So, I mean, thinking about them in a, integrated way is probably going to be useful going forward. But I have very little to say about it at this point because he's just now kind of getting his hands dirty with all of this data. So hopefully in the next few months, we'll have more to say on that.
That's amazing. Really looking forward to it. I just drove down those 7 hours down to Vanderbilt to meet with Katie, and we talked a lot about it. So very, very interesting.
She's the right person to talk to about this. She's been doing that work for years.
Exactly. Yeah. Well, thank you.
Thanks for your question.
Thank you. We're going to take an in-person question. Fidel, do you want to go ahead?
Yeah, thanks so much for a great talk. I have to admit, a big fan here. So you've got me sold, and I'm sorry about the controversies. I'm a big fan. I'm going to work as well. Department.
Same department. Come on.
Same department. I'm trying to navigate this conflict that I have.
Same soccer team.
I know. Related to the component number one and the relationship to breathing. And I've not had a chance to read your work in this last paper, so I don't know if the answer is in there. But any thoughts, or have you looked at whether that pattern is more maybe related to motion or changes in CO2, which relate to the level of vasoconstriction and what have you? Any thoughts along those lines?
Yeah, it's a great question. This original set of findings that's already published only looked at the Human Connectome Project resting state fMRI data. But what Taylor is doing now is getting all these other data sets that include all these different kinds of signals that may be underlying this global signal. And so he'll be better able to sort of answer this question, as I said, in a few months or so when we get our handle, get a handle of all of this. But for right now, it's it's just funny because we all know or think we know what we mean when we say the global signal, but it's very obviously a controversial pre-processing step in functional connectivity, whether or not to remove it. And so I think we've been stuck on the question for a while in our lab of, well, if you're going to remove the global signal, what is it you're doing? What are you removing? And of course, there's motion artifact in there, and there's scanner things and whatnot. But we know there's some neural component. We know there's some vascular. We know there's some ventilation. But it's hard. In fact, up until this point, I don't know if anyone can tell you exactly what proportion of all of those things make up the global signal. So we're hoping that some of these multimodal data sets with a lot of additional measures. Some of them are provided by Katie Chang's lab. They've collected a lot of these data. We're hoping that pulling all of this together might get us a better sense.
Thank you.
Thanks.
I'm going to ask a question first. So I was just curious, I know you mentioned on one of your slides how much variance is explained by the three components. And I think it added up to around 30%. Can you talk a little bit about the unexplained variance and what your thoughts are on that in terms of, is there something more there to probe but the tech isn't out, or yeah, I'd just love to hear your thoughts about that.
Yeah, the unexplained variance is, yeah, it's a bit of the signal, and I guess that's good. I mean, we speculated that it was maybe related to actual signals of interest, related to cognition or related to, something that the brain is doing. Because of course, these are all the resting state signals that are supposedly not related to task or not related to, a cognitive process. But yeah, one of the reviewers asked the same question, what do we think is going on with the rest of the signal? And I think, we don't have any way of making a good answer for this at this point, other than saying, that might be the signal that we most of the time are interested in, or that we most of the time focus on, the task related cognitive, processing and things of that nature. But I can't say with any certainty what that's about at this point.
Thank you. Andre, go ahead.
Lucina, great to hear your talk. Something that I guess thought about when I've read through the QPP literature is, of course, how there's a lot of studies that relate it to different traits, right? Whether it be like autism or ADHD or whatnot, but I've never seen any investigation of it in task fMRI, right? So I guess my question is, do these QPPs, are they preserved during the task evoked response, right? Should we be imagining like evoked responses happening atop the QPP, or are the evoked responses like disrupting like QPP2, right? Or does when an evoked response happen relative to a certain aspect of a QPP's time course affect the actual hemodynamic responses? Like, how should we relate those to each other?
Yeah, that's a great question. I'm going to reveal some of my ignorance here by saying that I think Shella Kilholtz's lab has done this kind of work, but I don't have the, summary of that off the top of my head. But I know she's looked at these patterns in animals as well, rats using, electrophysiology and stuff like that. So I think that they have done some work that is relevant to your question. But just from the cognitive neuroscience literature, we've, over the however many years we've had of doing task fMRI, we know that the evoked signals only account for some really small fraction of bold signal change, right? Like it's always been a very, very, whatever, less than 1% of signal change when you do some task. So I wouldn't be surprised if, the task stuff is just a small blip in this larger wave of things, what do you call them, QPPs or other large signals. I think the tasks are there, but maybe very small in terms of their magnitude of signal change compared to all this ongoing wave pattern.
So follow up, people, I guess, use the 20 to 30 second window for doing this. But might it be, I understand the brain's not changing drastically in a certain way, so that's a pretty good window, but might there be other states where it might be useful to actually change the window length, right? Like maybe sleep has different, yeah, you see what I'm saying? And how would you go about, I guess, figuring out how you're choosing the right window? Because, we could pick any window and find conceivably some pattern. Of course, these map onto other convergent findings from their converging across technique. Anyways, yeah.
Yeah, I think, I think we did a number of windows, and I'm trying to remember this, but yeah, you're absolutely right. Like deciding on the window length is non-trivial. So I think we did sort of more than one here, just as in some of the previous work. But yeah, I mean, it's kind of a chicken or egg question. Like if we knew what the right window was to look at, we wouldn't have to do it, but so I guess it's always sort of a little bit of trial and error with that.
Question?
Thank you for the great talk, Lucina. This is Lloyd. I was wondering if you're using any spatial dimensionality reduction. So when you examine and characterize the waves, is this done on a per voxel basis, or is there a coarser organization?
Sorry, is it on a voxel basis, or what was the last part?
Or like a coarser organization, like region or
Oh, sorry. Yeah, this was at the voxel level. Yeah, sorry if that wasn't clear. Yeah, so it's not been parcellated or anything. Yeah.
So okay, yeah, thank you. I guess I do have a follow-up question. Could complex PCA provide a spatial parcellation as well? Because many of these techniques, like ICA and PCA, they are used to get parcellation, but I understand that you're using that on the time dimension, so you don't get parcellation. But is it, maybe it's possible to extend?
Oh, yeah, that's a good question. Yeah, we hadn't thought of that in this study, as it wasn't sort of the goal. Jason Nomi in the lab is telling me this was done at the vertex level, so it's downsampled a little bit, but still, yeah, basically vertex level for the study. But yeah, I guess yeah, I guess in principle it could be used for that purpose as well. So yeah, we didn't sort of pursue that here.
No.
Hi, Lucina. It's Kalina Christo from UBC. Thanks for the talk. I just want to hear a little bit more your thoughts on this topic that came up about let's put quotes around the term for now, in the intrinsic negative relationship between the so-called task positive and so-called task negative networks. Let's call them default and dorsal attention network.
Sure.
So as we all know, we've heard, I think, some of the talks. There's this super famous paper by Fox et al. That claim that that's how the brain is organized, in these intrinsic anti-correlations. But as you pointed out yourself, and we've done some work about that as well, it's not as intrinsic as the term implies, and that might be dependent on the analysis and whether the global signal is removed or not. So I'm curious, what is your method tell you about that complex interrelationship and the intrinsicness or not about that anti-correlation?
Yeah, I mean, what's interesting is that the first principal component we see, resembles the global signal. The second one resembles a lot the test positive, test negative. So it doesn't seem to be just sort of driven, or it seems to be independent in a sense of the the first the global signal here. But I do remember these controversies going way back to the early 2000s. And I think in the very first paper I published on the default mode network in 2009, the reviewers asked us to do it with and without global signal regression, and we did it, and all these things. So I think a lot of us have probably noted over the years that the global signal regression certainly affects the findings in terms of strengthening in the sense, or pushing the negative correlations in a mathematically. But as far as like the intrinsicness of the relationship, I think it I don't think it's a total artifact. These I hate the term anti-correlation, but I guess we're stuck with it. But I don't think the anti-correlation that at Fox and everyone talks about is completely artifactual, if that's the question, because you can see it in so many different ways, sort of regardless of the GSR, or irrespective of your stance on GSR, you can sort of see some of these antagonistic relationships with like electrophysiology and in animals, for example. So yeah, I mean, yeah, it's an interesting question.
I'm going to ask another question, if that's okay. So I don't know if this came up in your talk, but I'm just curious about zero lag correlations. Have you done any multimodality work using like EEG and fMRI? Is it really zero lag, or is it zero lag in balls? Just have you given any thought to that?
Yeah, that's a great question. I mean, it's I guess the field calls it zero lag, and we've decided that that's acceptable. But yeah, it really would take the EEG fMRI data, which we now have from Katie Chang, to determine whether that's really the case. And so hopefully more on that to come.
No. Go ahead.
I just wanted to make a comment on PCA methods, and going back to your question about what you do of the 70% of the variability in data. If you look in the face processing literature that's focused on processing images of faces, the pictures of faces using PCA. What you start to do is as you move down the line in the component structures, you start to identify features that are are representative of the individuals that are in the analysis. So something that I don't know if it's a waste of time or not would be to start looking at the individual features of individual networks of those 50 subjects that were included in the analysis. And then see if some of those features are represented in 70% that's currently unexplained. I just want to throw that out there as an idea and get some people's feedback on whether they think that's a viable or is it, am I really actually crazy?
No, that's a great point. I think one of our reviewers asked about the consistency of these maps across subjects. So, we did this with a kind of a small sample across a bunch of different analysis, methods and approaches. But if in our small samples, the 50 and 50 replication, we did see a great deal of consistency, but you can imagine that with the thousands or 10,000 subjects or with different age development and different ages and things like that, you might start to see that that's where all the difference comes in. So I think would be a great follow-up.
Thank you. We have time for just one more question. Thought or question? Okay, well, thank you, Lucina. That was a really nice talk and a very interesting discussion. I am retiring as chair. Randy to finish, tell us what's going to happen next.